Hi, my name is Golia. I'm a PhD student at the Network Neuroscience Lab at McGill University. Today, I'm going to share with you findings from a recent project we worked on with Vincent Bazinet, another PhD student in the lab. Here, our aim was to investigate the spatial patterning of atrophy in frontotemporal dementia, specifically in behavioral variant of frontotemporal dementia. We asked whether brain network architecture and local transcriptomic vulnerability affects atrophy in FTD. And the relationship between connectome and brain disorders have been previously studied. The connectome architecture or the connectivity of the network is generally useful in the sense that it promotes interregional signaling and supports molecular transport among connected elements. Despite many benefits for communication efficiency and resource sharing, network systems are also vulnerable to damage, such that connections among elements allow pathological perturbations to spread between multiple nodes. For example, the schematic here shows how a single perturbation, which is indicated by the arrow here, can develop into a global cascade through the connected elements and reach distant parts of the brain. This concept has been used to study the transneuronal spread of pathology in multiple neurodegenerative diseases. One example is this work here, where the spread of misfolded proteins is modeled in Parkinson's disease, predicting the real atrophy patterns as a result of alpha-synuclein spread through the connectome architecture. The resulting patterns of atrophy resembled the underlying structural and functional network architectures in this study. Similar results have been shown for other disorders, where, for example, in this study, they showed that brain matter atrophy in multiple diseases resembles the spontaneous functional connectivity patterns and structural covariance networks. So brain areas that display high functional coactivations and have increased structural covariance tend to atrophy together, suggesting that spread of pathology is constrained by the connectome architecture. Altogether, the network and its topology may shape or influence the onset, the propagation, and the expression of a disease. Here, we wanted to test this hypothesis in behavioral variants of FTD and ask how regional vulnerability, in addition to the connectome architecture, contributes to atrophy patterns. First, to measure brain atrophy in FTD, we use deformation-based morphometry or DBM to estimate local changes in brain tissue volume density using T1 weighted MR images obtained from 2 FTD cohorts, a genetic cohort from GenFi dataset, and a sporadic cohort from FTL-DNI dataset. We then used linear mixed effect models to obtain group-level atrophy maps that quantify atrophy in FTD relative to healthy controls, while we controlled for age, sex, and acquisition site. Here are the results for both datasets. So in both datasets, we observed high atrophy in frontal and temporal areas as expected in frontotemporal dementia. The next question we asked was whether the observed atrophy patterns are focused to or are more pronounced in certain subnetworks or systems. So we first looked at the mean atrophy in 7 resting state functional networks. We compared the mean empirical atrophy values to a null distribution of means generated by a set of null models where the data was randomized, preserving the spatial autocorrelation of the brain. The results indicate a high atrophy in limbic networks, which includes frontal and temporal areas. We also investigated atrophy relative to the cytoarchitectonic classification of the human cortex, according to the classic von Economo atlas, where we observe high atrophy in the insular cortex and low atrophy in primary sensory regions. So altogether, all these shows that FTD-related atrophy appears to accumulate in specific systems that have certain functional roles or specific cellular compositions. So the fact that atrophy is more pronounced in a specific system suggests that atrophy is conditioned by the network architecture. If this is the case, a node that is mostly connected with high atrophy neighbors should display greater atrophy compared to a node that is connected to healthy neighbors. To test this, we measured the relationship between a node's atrophy and the mean atrophy of its connected neighbors. Here we defined neighbors as areas that were connected to the node with an underlying structural connection where the structural network itself was obtained from an independent data set from healthy individuals. We observed a positive relationship between the two, between the nodes atrophy and the weighted mean neighbor atrophy, when we use the strength of structural connections to weight the atrophy of the neighbors. We also find the same relationship when we use functional connectivity to weigh neighbor atrophies. The relationship was significant when we compared the empirical value to a set of null correlations obtained using randomized data with preserved spatial autocorrelation. And it was consistent when we repeated the analysis for another parcellation for both the structural and functional networks. We obtained the same results for the GenFi dataset as well. So a positive relationship between node and neighbor atrophy, which was significant in different resolutions against spatial autocorrelation preserving nulls. So all this shows that atrophy in FTD reflects the brain network architecture. Now that FTD-related atrophy is related to the connectome organization, we next asked which brain regions might act as the epicenters of the disease. Our hypothesis here is that a high atrophy node that also has high atrophy neighbors is more likely to be an epicenter compared to a node with relatively healthy neighbors. To test this hypothesis, we ranked the nodes based on their atrophy and the atrophy of their neighbors, and identified nodes that were highly ranked in both lists as the disease epicenter. So the results of this data-driven approach are shown for both datasets here. And what we see here is that the areas that are most likely to be an epicenter are mainly located in the insular cortex. To validate the findings of the data-driven epicenter analysis, we use an agent-based spreading modeling approach. Using the susceptible infected removed, or the SIR model, that simulates the spread of pathology across the cortex. Pathology, which can be, for example, a misfolded protein, propagates via the structural connections between brain regions and induces atrophy both pre and post-synaptically. So here, using this model, we simulate the transneuronal spread of pathology from seed regions through structural connections and estimate the correlation between the observed, or the empirical, and the simulated atrophy patterns. The simulated based most likely epicenters are then defined as seed regions that result in largest correlations. So for example, the model fit and the largest correlations for each seed regions are shown here for FTL-DNI, resulting in high correlations when we use insula, superior frontal, and lateral orbitofrontal regions as seed regions, with insula being the highest one, giving us the highest correlation. So this confirmed the results of the data-driven approach, and we find that the largest correlations is obtained when insula is used as the seed region. And we can even look at the empirical atrophy and the simulated atrophy patterns, and we see that the two are significantly correlated. And we observed a similar results for the GenFi dataset. Finally, we assessed the potential contribution of FTD-related genes to the simulation-based propagation of atrophy using mRNA transcription data from the Allen Human Brain Atlas. So we generated maps of regional gene expression for 4 genes that have been indicated in behavioral variants of FTD. The gene expression maps were incorporated into the agent-based spreading model, or the SIR model, controlling the synthesis and clearance of the misfolded protein. We then rerun the model with the regional heterogeneity from these gene expressions and compute the correlations between empirical atrophy and simulated atrophy patterns with the insula selected as a seed region, because insula gave us the highest fit, the best fit in the previous analysis. So the maximal correlations for each gene were compared to the maximal correlations obtained from null distributions, controlling again for spatial autocorrelation in the data in both data sets. In both data sets, we find that adding regional heterogeneity for synthesis and clearance of C9 or F72 and TAR-DBP genes improves the model. So in summary, building on previous research, the findings here demonstrate that atrophy pattern in BVFDD is conditioned by connectome architecture. Local transcriptomic vulnerability contributes to the observed atrophy patterns. Network atrophy appears to be centered at the insular cortex, and association between atrophy and connectome architecture is consistent in genetic and sporadic BVFDD. So with that, I want to thank you all for listening and thank everyone in the lab who helped us, the collaborators, specifically Vincent Bazinet, also the data consortia, FCL, DNI, and GEMPI datasets. This study is now published in Brain. So if you want to read further, it is available here. We also have a poster. If you want to learn more, you can stop by at our poster. And here is our contact information if you want to send us an email with your questions. Thank you very much.